Hello friends, I am Dr. Sachidanand Singh. I am professor in biotechnology. I am very happy to teach you all the topic structure determination and molecular modeling under the concept of NPTEL and SWAM course of IGNOU that is on protein structure analysis and determination. So, the topic which we will study today is structure determination and molecular modeling. Why do we study structure determination and molecular modeling is we have to understand protein structure prediction and we want to understand the prediction of protein. So, to understand protein structure prediction and molecular modeling, all the scientists around the world are moving ahead to understand protein structure in different ways. Why we want to study protein structure? The main focus to understand protein structure is to understand the function of the protein. There are different ways to understand protein structure. One first way is through sequence, second way is the secondary structure, and third is through tertiary structure. So, protein structure prediction and molecular modeling approach is utilized to understand protein structure prediction and molecular modeling approach. Under protein biological workforces, it is very important to understand from DNA, that is functional unit of life, to understand protein. Why protein? Because protein is involved in n number of functions. Protein is involved as a enzyme activity, protein is involved as transporters, protein is involved as drug action, protein is involved as target proteins. So, to understand the protein structure, its active site, it is very important to understand its structure. That is why the module here we are trying to understand protein structure prediction. To start with, a protein has a sequence, that is a linear amino acid. After this sequence is converted into secondary structure by the help of fold is your secondary structure prediction, which includes alpha helix, beta sheets, loops and coils. When these again gets turned up, when they again gets wind up, the protein is converted into tertiary structure or quaternary structure, which has active sites, and then the function is being produced. This is what a structure or a graphical representation of sequence to structure to function. This slide represents the 3D structure of a protein, encapsulates two combination. The first combination is secondary structure, which is alpha helix, beta sheet, and the black line which we see is the turn, wherein the second one are supercoiled structure, which is a combination of secondary structure, that is combination of beta sheets in forward and reverse direction, combination of beta sheet with the loops, combination of alpha helix and beta sheet together. The biggest challenge of protein structure prediction is when we have the biological relevance of any sequence, and when we want to see the function of that sequence, it has to be converted into its secondary structure and then tertiary structure. Then only we will be able to understand the function. So, to overcome of this challenge, there are different methods of structure prediction. The future lies of protein is on the basis of this, that is from sequence to structure to function, and this all is encapsulated in a cell. Hence, it is very important to understand the structure of a protein. There are different methods to understand the structure prediction techniques in experimental and computational way. This slide depicts structure prediction techniques with experimental and computational method. There are different categories of experimental methods. The first one represents cryo-EM technique, which has few advantages and challenges. They were able to depict nearby structures which was less than 10 angstrom. The second one is single particle analysis, which includes a high resolution structure prediction, but it was able to identify only up to a 40 kilodalton structure of the protein. The third one and the fourth one are the present structure prediction techniques of your 3D electron and ED method, which which is been involved for structure prediction methodologies, and these are some techniques which is still going on. The most prominent experimental method of protein structure prediction is X-ray crystallography and nuclear magnetic resonance. X-ray crystallography method is the best method when a protein is being crystallized, then the 3D structure of that is being depicted out, wherein a nuclear magnetic resonance NMR method is a method of a solution based structure in which the protein structure is being presumed. Out of this whole, there are still so many proteins for which these all methods are not able to identify the structure. So, in that need, there is a high demand of computational methods to understand and develop the structures of these proteins for which we are not able to depict through experimental methods. So, these proteins can be developed computationally only by using the base of these experimental methods and take up for computational analysis. So, there are different computational methods to perform structure prediction methods. Here, protein structure prediction methods is been divided into two methods. The first one is secondary prediction method and the second one is tertiary structure prediction method. The secondary structure prediction method includes Chow-Fasman rule and different algorithms, wherein the tertiary structure prediction methods include three categories, that is homology modeling, threading and ab initio modeling. So, let us take the first secondary structure prediction method. There are different secondary structure prediction methods to depict secondary structure of alpha helix, beta sheets, strands and other method. These elements from during the early stages of the folding process has been applied to understand secondary structure. Knowledge of protein secondary structure regions along the protein sequence is a presentation to model the folding process. Helixes and strand structures are maintained by evolution, and the formation of regular secondary structure is an intrinsic feature of random amino acids and sequences. Improvement of secondary structure prediction is relevant and interesting because secondary structure prediction allow for a wide variety of conclusions on the fold of classification and function of protein, and in process provide important information for 3D structure prediction. Among this, the best methodology used for secondary structure prediction is Chow-Fasman rule. So, why we predict structure? We can label proteins by dominant structures. Protein classification and structural blasting includes different combination of secondary structure, that is alpha helix, beta sheets. You can see beta sheet and alpha helix together, combination of alpha helix in forward and reverse, and combination of alpha helix and beta sheets together. Secondary structure prediction is a very important aspect to understand different components and in different scientific objectives. The most important objective to understand secondary structure is the next level of identification of 3D structure, the epitope designing in vaccine designing process, the docking and drug designing aspect of understanding the cavities, and to understand the mutations also secondary structure plays a very major role. So, understanding secondary structure, a different type of questions can be raised in scientific community. Among them, few questions like what are the factors that drive a set of residues to form helix, strands or coil. So, understanding the amino acids which play major role in helix, amino acids playing major role in strand, amino acids playing major role in coil may depict a significant role in understanding a disease or a significant understanding of a ligand molecule. There may also be a question that are some residues more likely to form alpha helix or more likely to form beta strands or other. These all questions when arise can play a major significant role in understanding secondary structure prediction. By understanding secondary structure, now we come to a method known as tertiary structure prediction methodologies. So, there are different computational methods which are used for prediction of proteins, in which the first one is single alignment strategy and second one is multiple alignment strategies. So, the first presentation here representing is computational method used for prediction proteins as phase 1. So, what is there here is the flow chart very particularly explains that a sequence which is of interest, this sequence has come out of an objective of when a student has done a soil based analysis and he or she has got some novel sequence by sequencing strategy. That sequence is unknown. Now, to understand that sequence function, its structure has to be identified. So, for that there are different methodologies to understand the tertiary structure. So, the unknown sequence which has been identified, for that a template selection has to be done. After template selection, the alignment of the unknown sequence and template has to be performed. After performing this alignment, a model building is been done. This model is a tertiary structure which needs to be evaluated. If the model doesn't aligns or doesn't passes the Ramachandran plot or evaluation of protein modeling, then this step has to again go back and again the best template selection has to be done. So, this is as no we can see, if the model is not okay, we have to go up and we have to again repeat the process. If the model is okay, if it is validated, we can end the process and report the novel protein with its function. The next method of computational method used for prediction of protein structure is when we have not a very high similarity. It's again a same process where we have an unknown sequence of interest which we want to depict its function. So, to understand its function, the 3D structure of the protein has to be identified. For this, the protein sequence which we have unknown protein sequence which we have identified, for that again alignment and search has to be done, but this search when it has been seen, it does not have minimum criteria of search methodology, that is of 30%, hence database searching has to be done. When database searching is done for the whole sequence, part alignment is been identified. When part alignment is there, again the process will go ahead with model building. When the model is been built, loop modeling will be done. Loop modeling is nothing but the sequence which is not matching with the known sequence and the database search. For that, the algorithm itself develops the sequence as a loop. After developing this loop and the model, again the evaluation has to be done. If it passes the evaluation, the model is perfect, else it has to go back to database search and another best alignment short search template has to be identified. So, this was all about protein structure prediction on the basis of secondary structure and tertiary structure. So, my dear fellows, what we understood right now was secondary structure prediction and tertiary structure prediction. When we have an unknown sequence, we can go as different methodologies and different terminologies. One is to understand secondary structure in sense of alpha helix, second one is beta sheets, third one is loops and coils, wherein for understanding these again binding and their winding up, we need to understand the tertiary structure. So, for tertiary structure there was 3 methodologies. First one was homology modeling, second one was ab initio method and third was threading. So, for doing all this, we performed secondary structure and tertiary structure methodology. Now comes molecular modeling. So, what is the definition of molecular modeling? Molecular modeling is a powerful methodology for analyzing the 3-dimensional structure of biological macromolecules. There are many ways in which molecular modeling methods have been used to address problems in structural biology. So, what are the approaches of protein structure prediction as molecular modeling? The target is the sequence, and this sequence is what? It is a protein sequence. So, we can see the protein sequence has been reported, and for this protein sequence, what are the 3 methods which we are utilizing is known as molecular modeling approach. So, the first one is homology modeling, second one is fold recognition or threading, and third one is when we don't have any type of matching is known as ab initio method. So, for homology modeling, what are the steps? For homology modeling, minimum 30% similarity should be there. For threading, we should have minimum of small matching patterns within for the whole sequence, wherein when we don't have any matching at all for the sequence, we go ahead for ab initio method, which is also known as scratch based method. So, this presentation clearly depicts the approaches of protein structure prediction method at very one site level. If we can see homology modeling, what are the 4 steps? Threading 4 steps and ab initio 4 steps. Finally, the protein model which we have received, where we have to take it up. So, for homology modeling, first is sequence alignment, second one is identification of the target sequence, third one is model building, and finally the model optimization, wherein in threading we can see, in threading we do the alignment and the pattern search of through different databases it has taken. Finally, these database searches is taken and again a assembly is been done and loop modeling is performed to obtain the best optimized model, wherein in ab initio method lot of physics and chemistry is been utilized to do a attachment and making of one amino acid assembly and seeing the torsional angles and dihedral angles to attach to them and construct the whole 3D structure, and then finally doing a model optimization. Out of these 3 methods, the protein models which we have retrieved can be validated using different optimization tools and databases. When they pass this evaluation methods, they can be submitted to protein MDB databases. Protein MDB data, PMDB databases, protein submitted then goes to different algorithms and different databases for competition. One of that prominent one is CASP. In CASP, different competition happens and the best algorithm is being reported. So, now let us take phase by phase molecular modeling approaches for protein modeling. So, the first one is which one? Yes, homology modeling. Say in homology modeling, the presentation reports in a very specific way. We have a query sequence. This query sequence is an unknown sequence for which there is no structure. If structure is already depicted in protein data bank or in 3D structure, then there is no use of doing this whole exercise. Hence, a student, when they want to understand a structure of a sequence, first they cross verify whether structure is present or not. Hence, this is a query sequence which is already been seen that for this structure is not there. So, this query sequence will go into a process in which for homology modeling, which we already saw in the previous slide, minimum 30% identity should be there for going for homology modeling process. So, if this sequence passes 30% identity of a template, then through computational approach a whole structure will be produced, obtained and will be validated. So, now in the coming slide we will see how homology modeling will be performed stepwise. So, this slide very clearly represents the steps obtaining in homology modeling. There are different steps phase by phase how we do homology modeling. So, the very first aspect is target sequence, which is of point of my and your interest. Second, for this target sequence has to go in 2 different methodology. First, it has to go to UniProt. In UniProt database, this target sequence as an amino acid has to given as an input, and when it is been seen that there is no 3D structure for that, this becomes our question. Then this sequence has to be submitted into BLAST, basic local alignment search tool. In basic local alignment search tool, there are 6 phases of BLAST in which we have to perform with PDB BLAST, which is known as PBLAST. In PDB BLAST, the sequence which has been submitted, we will go and do a search with PDB. What is PDB? PDB is Protein Data Bank, which has been maintained by Rutgers University. In PDB, all the 3D structure protein performed by X-ray crystallography and NMR is being submitted. So this target sequence will go as a search in BLAST with PDB BLAST. When PDB BLAST is done, all those sequences which is greater than 30% identity will be given as a results. So then out of those we will identify which will become the best template, hence template recognition will be done. The template which has been identified significantly, that template and target will be aligned, will be mapped. So this is my target sequence, this is my template, these will get aligned. When this alignment is done, we know amino acid sequence has a backbone of amino acid, that is your amide bond, CONH2 group. So that is your backbone of the amino acid. So in the alignment, the first thing will be backbone prediction or backbone alignment. After backbone alignment, the gaps which is still left out, that process will go with the loop modeling approach. With loop modeling successfully completion, it will go to a next step, that is side chain modeling. Side chain modeling is nothing but the functional group which has been present to have a better 3D structure confirmation. Finally, the 3D structure protein will be obtained, but this 3D structure protein has to pass the evaluation method. So the very prominent evaluation method is model optimization by Ramachandran plot. So using the Ramachandran plot, the amino acids will be fixed into different quadrants and will be able to see the presence of their allowed and disallowed region of the amino acid specificity. Finally, when it passes the Ramachandran plot, this model will be validated and evaluated. So these are the steps in homology modeling. After seeing the flowchart of homology modeling, this slide depicts very clearly different homology modeling software present as open source and purchased or licensed version, among which homology modeling softwares, the prominent one is Modeler, and there are different other softwares also which are graphical user interface one. So this slide represents different homology modeling software tools and databases. This slide depicts the time scale of homology modeling, that year by year how the homology modeling software and algorithm have passed from different scale and year, and different softwares and databases with different novel and new and highly precise algorithm are being applied to give the best possible protein structure. So if we see from down to up and in the horizontal, these are represented as year wise, and we can see at 2020 from starting of 1990s, we can see n number of softwares and tools which are been reported yearly, and they pass through CASP also, which we already saw the previous slide as a competition for comparative protein modeling. So the next protein modeling method, the first one was homology modeling, now the second one is threading. So when we do threading, when in a protein sequence which is our unknown sequence, and when it is been done with database search, we have seen that it doesn't lies with the 30% of identity or similarity. So for this approach, the unknown sequence is been matched with different databases to get part alignments. So if we have different part alignments, those part alignment which is matching as similarity or identity, those component also can be taken together and can perform multiple alignment strategy method to develop a structure. So for developing this structure, there is a multiple alignment method, then seeing the libraries, finally developing part structure and binding them with loops and coils, and then finally evaluating this model using Ramachandran plot. And if it passes through Ramachandran plot, or if passes through model evaluation, the protein is been reported as the tertiary structure protein. So we have seen the different methodologies of protein structure in two categories, that was homology modeling and threading. Now, in this slide, let us see the comparative comparison of homology modeling and threading. Very clearly, this presentation depicts the how homology modeling, in which there is a sequence, and for this sequence a template is been identified, wherein here in threading there is a target sequence for which there is a library of sequences to be mapped. Here there is only one template sequence, which is a structure, and on which the whole overlapping and the unknown sequence structure is been developed, wherein in the threading portion there are different libraries or part alignment structures on which the structure has to be developed. Finally, in both the portion the 3D model is been built. Here the thread target of different structures are been combined together with different loops and coils. Finally, a best fit model is developed in threading, and here a best modeled protein structure is developed, but for both best fit model or modeled structure, it has to go through model evaluation. Finally, after model evaluation, the structure may be reported. So, now let us see the third method of protein structure modeling, that is also known as ab initio protein modeling, or it is also known as scratch protein modeling. And when we do ab initio protein modeling, when there is no alignment, there is no structure available for the unknown sequence. So, for this approach, the target sequence, the sequence for which we want to understand the structure, has to go in different strategies. The first one is knowledge based potential method, which includes different physical and chemical methodology. It also includes profile alignment method for understanding the sequence of the unknown sequence as a target sequence to understand the structure. So, the very first one is Monte Carlo fragment assembly method. Here comes different algorithm as Monte Carlo or molecular dynamics at later stage. So, here comes Monte Carlo fragment assembly method, and this includes, and this gives low resolution model proteins. So, this is all dependent upon the profile method and knowledge based potential. Finally, in the second phase, physical or physics based atomic refinement is done to see the each single amino acid combination. The structure identified by this has to again go for model evaluation. So, now we can see very clearly that the sequence which we have identified has to go either by threading method or either by ab initio method, and then clustering has to be done, that at central level, and finally the protein model which has the best E value, or the protein which has developed a best structure, has to report as through model evaluation and give us the best fit model through this approach. So, this was all about protein structure prediction by the three methods for tertiary structure prediction. That was what? The first one was homology modeling, second one was ab initio method, and third was threading. So, the model which is been evaluated as and passed through model evaluation has to also be a stabilized protein. It has to be a stable protein. So, to understand a stable protein, the methodology or the computational method to understand a stable protein is through molecular dynamics approach. Molecular dynamics approach is nothing but to understand computationally the stability of a protein in nanosecond or picosecond. It includes different algorithm methods known as Monte Carlo or molecular dynamics approach. This is a promising approach of protein modeling. So, here initial geometry of the protein is been taken up and different force field is been utilized. The force field are different, say for example SHARM, MMGBSA, molecular modeling, generalized bond surface area. These are different algorithms which are being utilized to stabilize the protein. Finally, energy minimization is done to understand the stability of the protein. Later, equilibrium strategies also are seen, as this protein is in our body and this protein also moves as NTPK. this proteins also has to pass pressure and temperature. So, equilibrium also has to be studied. Finally, the analysis is done using the different algorithmic approach of Monte Carlo and molecular dynamic approach. This is known as post processing in molecular dynamics. Hence, this was the whole alignment, whole understanding of a protein sequence to a secondary structure, to a tertiary structure, and finally understanding the stability of a protein as through molecular dynamics and Monte Carlo approach. So, in this whole video, what we have understood is protein modeling and molecular modeling approach. So, to concise the whole learning approach, let's see and conclude this. In the first phase, we have understood the importance of protein structure prediction and molecular modeling. Second aspect what we have studied is different types of experimental and computational techniques for protein structure prediction. Third phase is primary secondary structure of protein has been analyzed and studied. Stepwise different methodology for molecular modeling of protein is been understood. Protein stability testing and analysis using molecular dynamics was been studied at the later slides. Next, in the coming video, we will study what are the different softwares and databases for performing comparative protein modeling techniques. Thank you.